Okay, so now we're going to focus on the second line of defense, which is basically your cells that are pretty much like phagocytes, phagocytic cells. It also includes any kind of like antimicrobial chemicals that include things like, like your antimicrobial peptides or the complement, which we'll look into, which has lots of different components to it, hence the name complement cascade, and even things like interferons, which are specifically useful for things like viruses. And then, of course, so in addition to, so hold on, I'm going to start doing this. So the second line, I've got like smaller markers, so hopefully this will be better. The second line, we're going to see things like actual cells that are involved, like phagocytic cells. We're going to see that there's going to be chemicals involved, both made by those phagocytic cells. Don't necessarily have to just phagocytosize something. They don't necessarily just eat something. They can even release chemicals, and we're going to look at a little bit of those like phagocytic chemicals and even like the non-specific chemicals. And not by phagocytic, I mean like the chemicals made by phagocytic cells. And then we also have like chemicals like the antimicrobial chemicals, like antimicrobial peptides, complement, interferon. So if we just use that, actually that would work. Antimicrobial chemicals, interferons, and then complement, right, which is part of the complement cascade. So that's all important. And then another defense mechanism of the second line of your innate immune system are just various processes that your own body does. And these processes include things like inflammation, which I know does not sound fun. And in fact, acute inflammation is great. Chronic inflammation, not so great. That can cause severe damage, even death to tissue. So we just want the right amount of inflammation, because that does have really like positive effects. And then, of course, fever, which is the same thing. The right amount of fever is really good. Too high of a fever, that could be quite detrimental, and can denature proteins. It can, cause. It can like even like thingamabob like nerves, and so it can lead to things like hallucination, coma, and then eventually death. So small amounts of these things are good. Big amounts of these things, like too high of a fever or too much like chronic inflammation, are bad. So this is what we're going to be looking at. So one thing before we actually do the full, spider web, blood is really important when it comes to, from here on out, it's going to be really important. We're going to talk about a lot of stuff with it. Okay. And so your blood's actually made out of two different things. We have the plasma, which is just basically like the fluid. So the fluid that contains like the water, basically, that contains electrolytes such as ions and gases and nutrients. And then we have what we call formed elements. And in these formed elements, that's where your cells come in. So your red blood cells, your platelets, your white blood cells. Okay. So for your plasma and everything, it's really important because it does play a part in inflammation. So it does play a part in inflammation. It does also play a part in, it has blood clotting factors. So it has all these proteins and everything inside of that helps. And in fact, actually, the plasma without blood clotting factors is known as serum. So plasma minus blood clotting factors is equal to serum, which we've talked about a lot in like diagnosing infections. The plasma also has proteins like ferritin or transferrin, proteins that sequester iron. And I don't know if you guys remember this, but a lot of bacteria actually need iron, and they have specific methods to get the iron. So some part of their virulency is actually to get like methods to get iron. So some bacteria have siderophores, which go and like seek out iron in the blood. Some bacteria can lyse. Remember hemolysis, so beta hemolysis, they can lyse red blood cells and attach, like get the iron just like that. But iron is something that a lot of bacteria need. There's some bacteria that don't, like Borrelia burgdorferi, but for the most part, iron is something that they need. You also find complement proteins in here. I'm just gonna draw it right here. So proteins that are part of the complement, and so that's right here, right? It's part of that complement cascade. So like C3, and we'll talk about the proteins, those complement proteins in just a bit. You also are going to find, and these are more things for the adaptive immune response, but you're going to have immunoglobulins and so Igs, immunoglobulins, and antibodies. Yeah, so you have all sorts of things that are found that are necessary, like proteins that work as like, you know, like as barriers, so like sequestering the iron would be kind of like a barrier, and like providing help to the actual immune response, like complement. Even blood clotting can also provide help in different ways. I will just see soon. Okay, and then we have our formed elements, which includes things like red blood cells, right? Or also known as erythrocytes. So erythrocytes, these are really important for oxygen delivery. We also have, I'm just going to draw it right here, platelets, which are really important for clotting. And then we have the ones that we're really looking into, like that we're really interested in, are the leukocytes, or basically your white blood cells. Okay, your white blood cells. So your leukocytes can actually be split into two groups, and the two groups are mainly there just for what they look like under the microscope, but like specifically what we find inside their cytoplasm. So their cytoplasm can either be granulated, like this has like different granules in it, or it could be like a uniform ungranulated cytoplasm. So that's what we split them into. We make them into granulocytes, which are obviously the ones, it's in the name, they have a granulated cytoplasm. And the other ones are known as agranulocytes because they don't have granulocytes. They don't have, like, they have a uniform cytoplasm. They don't have any granules in the cytoplasm. So let's look at the granulocytes real quick. We have basophils, which have a really cool name. I think they have a really cool name. These guys, basically, they release, like, chemicals necessary for inflammation. So they kind of, yeah, I'm just gonna say release chemicals for, so chemicals, for inflammation. They're also really important in a process known as diapedesis, or diapedesis. Not sure how you guys want me to pronounce it. I pronounce it diapedesis. And so basically with that, it's just, it causes the like cells to kind of squeeze through blood vessels. And so important cells like phagocytes can squeeze through the blood vessels and get to the actual localized area of infection. So it's a very important process. In fact, actually, all granulocytes do this. So including eosinophils, or eosinophils, it's probably how you pronounce it in English. These guys can actually go through phagocytosis. Okay, so they usually ingest their cells. They can, through the process of diapedesis, these guys can also exit the blood and kind of go to where the fight has to go. So they also go through diapedesis. They can, oh, they're very, okay, so it's interesting because if we have an increased amount of eosinophils in the blood, that usually means you have like a parasitic worm infection, fun, or an allergic reaction. And that's because they defend against parasitic worms. And that's why an elevated amount of eosinophils means that you have parasitic worm in you, can mean that. Parasitic worm, and they're also the ones that are present in the allergic reaction. Okay, so that's basically them. And then finally, the last granulocytes that we have are neutrophils. Neutrophils, all of these guys. Okay, so these guys are also basically, it's important for phagocytosis, and they also go through diapedesis. Okay, so they exit the blood and they can go towards the actual like localized infection and fight. And then we have over here our agranulocytes. So they're split into two. We have our monocytes here, Monocytes are just the immature version of macrophages. So they technically mature into macrophages, which is probably a word that you have heard a lot, and you probably remember that macrophages eat things up. So macrophages phagocytize the bad guys. So they phagocytize like foreign objects and everything. Sorry. And they also go through diathesis. I really should look up to see how you guys say this in English, and because I keep pronouncing it in Italian in my head, so that's bad. And then, yeah, so that's basically it for them. And then we have lymphocytes in the other corner right here. And here with the lymphocytes, we're not going to get into too much detail about them right now, at least. This is because they're mostly involved with the adaptive immune response, okay? But yeah, mostly involved with that. However, one lymphocyte that we are going to look into for the innate is the is the natural killer. Yes, there are those, that's their names. And so I'm going to, like a lot of times in books and everything, you just write NK. So natural killers can work with them. And these guys kill. These guys are pretty good. So that's basically it. I also do want to remind you guys of, oops, sorry, I just hit something. Dendritic cells. If you guys remember dendritic cells, we already talked about them. They can be found in the skin and in the mucous membrane, right? They're also they're technically not white blood cells. They're not, by technically I mean they're not white blood cells. They look really cool. They're really like branched out, like I can't do it. So they're very branched out. And they go through phagocytosis. Okay, and then they also informs your cells, informs your adaptive immune cell, adaptive immune response cells, and informs them of what to do. So really important, guys, as well. So these are kind of the players that we're going to be looking into right now. So I'm just going to do this, some of the players, and then we're going to look at the chemicals and the actual processes. So yeah. And again, I'll take pictures of this. But for now, let's keep going. Hopefully it's fresh in my mind, I guess. Yeah. Okay, so the second part of this is, again, the innate immunity. Second line of defense, so our cells, our chemicals, and just the various processes that we have. Okay, so first off, let's look at our cells. Let's look at those phagocytic cells. So I'm going to make a line over here. So phagocytosis is obviously one huge line of defense. My pen, unfortunately, is finishing. So we already kind of looked over the cells that do this, but just to kind of recap, right? We have things like neutrophils, and so yeah, granulocytes like neutrophils and eosinophils, however you want to pronounce it. You also have some agranulocytes like macrophages, really important. And then, of course, you have, outside of the whole white blood cell things, you have your dendritic cells as well. That go through it just flew away. Go through phagocytosis. One thing I'm not gonna talk about, but you can differentiate all of these leukocytes depending on what they look like under the microscope. I'm looking at their the cell itself with the nuclei, not the nuclei, sorry, the nucleus. They all look kind of different, and that's how you can readily diagnose them or see them, or observe them. That's the word I'm looking for. But I'm not gonna. That's not really that important. So, not for this case, at least. So phagocytosis, what's happening? You're eating up your pathogen. And so there's a couple of steps that need to occur in order to, for a phagocytic cell to go and eat the pathogen, right? So the first one is, the first step, of course, is to actually go to or be drawn to your, the foreign object that you want to get rid of. So usually this is known as positive chemotaxis. So it's going positive. Oh, I spelled positive wrong. I forgot the IVE. Positive chemotaxis. So a chemical or like the foreign object, you're positive because you're moving towards it. Taxis is movement, so think of taxis. So it's movement of the cell basically towards the chemical stimulus. And they move through like different pseudopods, like they extend the pseudopods, and that's how they move towards the microbe. And they can be attracted to all sorts of different things that can result in positive chemotaxis. It includes things like microbial secretions. It could be parts of the actual microbial cells, part of damaged tissues. You can have chemotactic factors like defensins or various peptides that you get from the complement. You can even get like from chemokines, that are released by chemokine chemicals that are released by leukocytes. And you can also, they can even be attracted to like parts of like damaged tissue. So various areas that they get attracted to. After they get to the actual place, they have to adhere to it. So they have to attach. So it's called adhesion. So here they attach usually through some glycoprotein/receptor action. And this is where like bacteria that have capsules, one of the way they prevent themselves from being phagocytosized is because they have that capsule, the phagocytic cells can't attach to the actual bacterial receptors or whatever chemical they're attaching to, like a glycoprotein usually. So yeah, with adhesion, there's also another thing to talk about is that there's a process known as opsonization, which is when, if this is my bad guy, okay, that's my bad guy, lots of little like proteins will, like of the human body, can kind of surround the bad guy and make it kind of like this huge signal for a phagocytic cell to go and like surround and attach to the bad guy. And then it allows for a phagocytic cell to more easily identify and eat up the bad guy. So this is known as opsonization. The proteins that do this in general are known as opsonins. And usually this could either happen through things like your complement. So the complement proteins, which we're going to talk about, complement proteins can do this, and even like antibodies as well can bring about opsonization. So if you have opsonization, your phagocytic cells are helped pretty much. So once you have attachment or adhesion, then of course we have ingestion, right? And then once this little so this is the bad guy is eaten up, it actually gets put into a small food vacuole at the beginning. This food vacuole is known as a phagosome food vacuole. And then the phagosome in the fourth step is going to actually mature and like come together, fuse with the lysosome. So this is known as phagosome maturation. So matures into maturation. So the phagosome fuses with the lysosome, and if you guys remember, the lysosome has an acidic pH, right? It's basically the stomach of cells. So it has an acidic pH. It has digestive hydrolytic enzymes, so enzymes that break down fats, the enzymes that break down nucleic acids, so enzymes that basically get rid of stuff. And then inside of it, you also have like toxic species of oxygen, so like oxygen radicals, and it's all a way to basically destroy, right? You have like this, now the microbe, which is in the food vacuole, is going to be the food vacuole, the phagosome. It's going to lyse to the lysosome. So I'm not lyse, sorry. I keep saying lyse. Fuses with the lysosome. Okay, and so it's like the mini stomach of the cell. And so now we have things like digestive enzymes, low decreased pH, toxic oxygen species, all sorts of things like that, which are going to get rid, and this comes to the fifth part, which is killing the actual bad guy, right? These are all things that contribute to the killing of the bad guy. Again, a lot of times if your bacteria is capsulated, or if the bacteria has things like waxy material in its cell wall, so think mycobacterium, this actually gets prohibited. They're safe against this lysosome action. And then we have the killing. And so then you may think that this is where it ends, but sometimes it's not completely complete. So like digestion of the microbe isn't necessarily fully complete all the time, right? And so it doesn't always complete. And so if that happens, any kind of wastes or microbial like parts, I'm going to write here parts, get removed via exocytosis. So it's just basically the opposite of phagocytosis. It just regurgitates everything to the outside. And then here, these, yeah, they would get removed by exocytosis, and it actually kind of helps, like these small little parts. They basically, like, dendritic cells can attach to them and everything, and it can actually play a role in the adaptive immune response. So it can play a role in the adaptive immune response. Okay, so that is phagocytosis. Like I said, though, before, not necessarily do all of our phagocytic cells, I mean, even though our phagocytic cells can phagocytosize, they don't have only that to kind of protect themselves with. They can also do non-phagocytic things. So I'm going to make another line, like here, and just right here, I don't know how to write this, non-phagocytic things. Is that what I'm going to call it, non-phagocytic things. So they also do non-phagocytic things. So let's look at some of our, some of our phagocytic cells that do this. There's also things like natural killers that we're going to be looking at that, like, we'll see how they work, okay? So for example, eosinophils, right? Remember, they phagocytosize. They can also, especially towards things like parasitic worms, because remember, eosinophils, like they work against parasitic worms most of the time, or with allergies. They can literally send toxins, so they can attach to the parasite, in this case, the parasitic worm, and then release toxins to them. So they can release. So how do I write this? Attach to worm and release toxins. Usually if the microbe that they're handling is just too big to eat, right? So for big microbes, so can't eat them, they're going to release like antimicrobial chemicals are released. Okay. And then, yeah, another really amazing thing actually about these guys is that they can use their mitochondrial DNA, which is, I love DNA and microbiology. These are my two favorite things. And so, like, this just really excites me. So mitochondrial DNA, which has, amazingly enough, antimicrobial activity. Basically, the mitochondrial DNA of the eosinophils plus some other eosinophil, like, proteins, they can actually form a barrier, right? Which can like traps like microbes, specifically actually bacteria in this case. Actually, I'm gonna write that down. Bacteria, because it's the bacteria, like some certain bacteria that allow to activate the mitochondrial DNA in this way. It traps some bacteria and then kills them. So it's just very interesting how that works. Okay, so then which one are we going to talk about? Let's talk about another granulocyte, the neutrophils, because remember, basophils don't phagocytose. So let's do the neutrophils right now. Okay, so neutrophils, they have all sorts of really neat enzymes. Like, I'm going to write here, neat enzymes. And these enzymes can literally produce toxic oxygen species like the free radical oxygen or like hydrogen peroxide. They can also produce things like nitric oxide, which is really important for inflammation. Okay, really important for inflammation. They're really, poor guys, they're really neat. As they die, they actually start forming this weird, like, so the neutrophils are dying and they form this kind of fibrous net. It's literally, they're actually called nets because they stand for neutrophil, like, what is it, extracellular traps or something like that. And these nets, so as they're dying, they're forming these nets, these fibrous nets, and they actually trap the bacteria. So they immobilize them, and like they kind of sequester them, and they allow for antimicrobial peptides to to kill the bacteria. So they trap bacteria when they die. So like they go, even as they die, they fulfill their roles. When they die, so by they I mean when neutrophils die, they sequester them, immobilize them, and then they release antimicrobial peptides, and then we have antimicrobial peptides, which will ultimately kill the microbe. So pretty sweet if you ask me. Okay, and then last but not least, we are, like I mentioned before, we are going to talk about the natural killers. I'm going to, hopefully you guys can see that, where this line comes from. So we have natural killers, NKs. And natural killers, they basically, they secrete toxins. They secrete toxins and the toxins aren't like to actual like pathogens. They secrete toxins to infected cells, specifically virally actually infected cells, and even like tumor cells. So it's hard because they really can't differentiate between them. That's why like the faster they are, the better. Anyways, so they're programmed to basically like they're natural killers. So they're programmed to kill other cells, and they stop only when they receive an inhibitory signal. So stop once received an inhibitory signal. Hopefully you guys are working along with me, right? And you're not just looking, but you're making a web map, a spider web, a concept map of your own. Okay. Yeah. So should we talk about the processes next or the chemicals? Let's talk about, I don't know how to draw this. I think I'm going to run out of space, guys. This is bad. Okay, let's talk about the non-specific like chemical defenses. And by non-specific, it's just because they do work along, like this is the innate immunity. So it is non-specific. It works against a lot of like wide ranges of things, chemicals, non-specific chemicals. Okay, another circle right here. All right, so first off, there's a lot of like receptors that we have to talk about. Receptors that actually bind to these things known as PAMPs, or basically pathogen-associated like molecule, crap, what does a P stand for? Yeah, pathogen-associated, oh dear, I forgot. It's gonna come to me. Anyways, it'll come to me eventually, and I'll talk about it once it does. But this is basically the way, like these receptors are the way for your body to recognize self and to recognize what is foreign. So your body obviously doesn't want to hurt self, right? But it does want to hurt foreign material like microbes and everything. And so it's those particular patterns. Sorry, PAMPs. I got it, guys. PAMPs are basically stand for pathogen-associated molecular patterns. So it's those patterns of the bacteria or whatever microbe you have that are going to be received by these receptors. And then these receptors then go ahead and trigger, by binding, after they bind to these PAMPs, they go ahead and trigger various types of pathways, including things like inflammation, including things like interferons, which are actually chemicals of their own. Yeah, like other kinds of, even the adaptive immune response. Yeah, so how do I write this? Okay, let's write these things as receptors of PAMPs. Okay, so pathogen-associated molecular patterns. And you have a few of these receptors. Now, the first one, you may even have heard of it, I'm not sure, but toll-like receptors, there's a bunch of them. Toll-like receptors are found on the membranes. So they're found either on like the membrane, like the plasma membrane of a phagocytic cell, or even the plasma, the membrane of a phagosome. So remember back here, the phagosome, the actual membrane of that food vacuole. So they're found on membranes. Okay, and they once they bind to to the PEMPs, binding triggers. I'm going to write here binding triggers. It triggers all sorts of things. It triggers things like secretion of inflammatory or inflammatory molecules. Okay, so secretion of, this is good because this is like testing myself as well. Like seeing what I remember and like, you know, where I need to obviously work on stuff. So secretion, and I know you can't see, hopefully, maybe if I turn like this, it's better. Secretion of inflammatory molecules or mediators even, yeah, inflammatory molecules, or even like interferons, like they actually secrete interferons, and we'll talk about interferons in just a second. They can also make chemicals that stimulate the adaptive immune response, and it can also lead to apoptosis, which is basically cell death. Okay, so the apoptosis and cell death, but of which cells? Of infected cells. Obviously, we don't want no non-infected cells to die. Okay, so that's what toll-like receptors do. Another kind of receptors are known as nods, N-O-D, but these are going to be found in the cytoplasm of our cells instead of like the membranes of our cells. These are in the cytoplasm of our cells, and they also can trigger things like inflammation and apoptosis and other innate stuff. So right here, inflammation. So it triggers inflammation. I believe it also triggers apoptosis, and usually it does more stuff for the innate immune response. Okay, all right. So the next thing that we're going to talk about with non-specific chemicals are your interferons. So I'm going to make another line. I wonder if I should make my lines with this. No. If you follow along with me, this should make sense. So line right here, and now we're talking about interferons, IFNs. Now interferons are, again, non-specific. These are non-specific chemicals, part of the non-specific innate immunity, and they basically go and inhibit viral infections. That's their point. They're very like fine-tuned towards viral infections. Yeah, so how do I write that? So they non-specifically inhibit viral infections. There we go. Okay, non-specifically inhibit viral infections. As they do that, though, they do release, like that's what you see, it's an N. They do cause a few signs, like a few symptoms to arise. So symptoms like general, like discomfort, so malaise. You have body, like muscle aches, like myalgia, chills, headaches, and fevers. And if you're hearing these things, you're probably like noticing that these are typically the signs and symptoms of a viral infection, and that's mainly brought about by your own immune system bringing it up. So they do have, I'm going to write here, symptoms that it can bring up. Okay, so we have myalgia, malaise, headache, fever, chills, etc. That kind of stuff. Those are the only things I can think of right now. Okay, so there are two types of interferons, type 1 and type 2. I want to talk about type 2 first, just because it's going to fit in this little nook that I have here, and then type 1 is going to come here. So type 2, this is also known as interferon. I spelled IFN, not INF. I spelled the INF, sorry. Interferon gamma. Okay. Type 2, these are going to be activated by T lymphocytes. Okay, and even by natural killers, which, if you remember, are also lymphocytes. The only lymphocyte that we actually mentioned right now, but there's more lymphocytes, obviously, like T lymphocytes, B lymphocytes. We'll get to that in the adaptive. Okay, so These guys are activated by T lymphocytes and natural killers. T lymphocytes do take a little bit longer to activate themselves, so like to themselves be activated, and then once they're activated, they take a lot, like, you know, they then have to like stimulate the interferon gamma. So this can happen usually like days after the infection, so it shows up later because of that. I'm not gonna write that whole spiel out. I'm just gonna say shows up later compared to the type 1 interference, which we'll talk about in just a second. Outside, just hurt myself. And so what do these guys actually do? I'm gonna write that right here. They stimulate the activation of macrophages. So they're involved with macrophage activation. Macrophage activation, and because of that, they can be, obviously, like, macrophages can work against viruses, but macrophages work against everything. So it's kind of a way of regulating the immune system. So I'm going to write that actually down. They regulate, therefore, the immune system. Okay. So macrophages, they're not as virally like specific, I guess you could say. But type 1s are. Okay, so type 1, and here we have interferon alpha and interferon beta. Okay, so these are your type 1, and they are, yeah, they're a little bit, they can be brought about by lots of different kinds of cells. So for example, like macrophages, even monocytes, so even like immature macrophages, macrophages, and like even like various lymphocytes can secrete interferon A, interferon alpha, and then, oh my gosh, sorry, hold on. I apologize for my doggy. I have no idea what he was barking at. So they could be released by like macrophages, and what was I saying? Yeah, various lymphocytes can secrete, you know, things to get the interferon alpha, and then fibroblasts can secrete, can basically, you know, release interferon beta. So essentially, actually, they're released by cells that are themselves infected. Okay, so they act on the same cells that actually secrete them. Okay, so these cells are infected cells. I'm going to write here, cells infected by the virus. They secrete them. Crap, I shouldn't have read that. Yeah, I'm just going to say secrete. And then they act on them. Okay. And so once they do that, the cells infected by the virus, like, you know, they get to the cells infected by the virus, they can trigger these protective steps. And hopefully these protective steps are going to be towards the actual cell infected by the virus and not the cell surrounding the cell infected by the virus. I'm trying to see if, like, I don't know why you guys can't see this as well. It's kind of blurry, isn't it? So I'm sorry about that. But so they really try to make it only that the cells infected by the virus are harmed, but not your own cells, like your non-infected cells. We don't want that. So how do they do that? So they bind to the actual cells, and then they actually trigger like this protection. They could trigger the protection by making things known as AVIs. So AVIs, and these are antiviral proteins. So here, let me write antiviral proteins. And these proteins, pretty much, they are what they sound like, right? They're antiviral. They inactivate the viruses, usually by like they inactivate the virus DNA or something like that. Some antiviral proteins include like oligodenylate synthetase or protein kinase K, and these are going to specifically address things like in protein synthesis. So, for example, oligoadenylate synthetase actually goes ahead and destroys the RNA. And as you guys know, RNA is like that intermediate messenger that's needed to then make proteins out of it in transcription and translation. And then protein kinase R is going to actually inhibit the synthesis of proteins by ribosomes. And so all in all, they basically destroy the ability for that particular infected cell to make proteins, and therefore of course, the virus can't, like the virus's proteins can't be made, and so the virus stops. It's no longer made. So that being said, this doesn't mean that your own cellular metabolism, like the metabolism of your own cells, doesn't get affected. Of course, it gets affected. Basically, that infected cell is done for. Yeah, but you're trying to, you basically sacrifice that cell to get to the virus's component, and to prevent the virus from making, or from hijacking your system to make proteins. Okay, and they can also even activate natural killers and other lymphocytes, part of the adaptive immune response. Okay, and with these guys, they simply kill the cells, kill the infected cells with the virus. They kill the virus infected cells. Okay, boy, I am hungry. All right, so I guess the only positive thing about recorded lectures is that I get to take a break and have a brownie in between. So, yay. All right, what was I talking about? Where are we? So non-specific, we've talked about the receptors and the PAMPs. We've talked about interference. Yeah, yep, okay, so the next thing, this is a bit big, little bit long, is the complements, okay? And if you remember, it's actually part of your plasma. It has complement proteins. Okay, so now we're going to talk. Oops, runaway pen or runaway marker. We're going to talk about the complement, and I'm going to make it all the way down here. Yeah, so I'm going to do this. Oops. Complement. Here we go. So basically, the complement, all in all, what it does is just it destroys foreign cells. It could do this in a variety of ways. It's going to destroy foreign cells by either by of course causing opsonization, because they themselves act as opsonins, which we talked about with how it helps phagocytosis. This is why I really love concept maps, because you can see everything connect, right? So they can act as opsonins. They can release chemotactic factors, and they can also cause inflammation. Like they can trigger. I don't know why I wrote trigger. Dang it! I should not have written trigger. Inflammation and fever, right? So all things that will ultimately cause a destruction of your cells. And so yeah, they usually act really quickly without destroying your normal cells that are neighboring the pathogen. They usually just destroy the actual foreign cells that are the pathogen itself. So they usually don't. They act rapidly against the pathogen. So how do I write that? Should I write that? I'm just going to do this, like a little bracket that encompasses everything they do, and say, this is away from the interference, and say destroy foreign cells rapidly, and not your own cells. Okay? All right. So what is the complement? Complement is basically like this cascade. It's known as a complement cascade because the complement cascade, I'm going to do it. I'm going to do it over here. Complement cascade. It's known as a cascade because literally like one thing's activated, and then the product of that particular reaction is going to activate the next reaction, and then the product of the next reaction is going to activate the next reaction. It's basically like a cascade of like a domino effect, essentially, of like the sequence of chemical reactions where the product of each reaction catalyzes the next reaction. And so in it, you're going to see like lots of cleaving, like there's specific things that are together, right? That like say two subunits that are together. And then once you cleave them, one of them becomes active and it goes ahead and does something else. There's lots of cleaving, there's lots of fragmentation, and these fragments, they can become active, right? And the fragments themselves can really help. So what do the fragments do? They form fragments. You can have fragments that will combine to form new enzymes, combine to form new enzymes. So enzymes either that are necessary for the immune response or enzymes that continue the complement cascade. They can also enhance inflammation. So I'm just gonna write inflammation. I keep write like enhance or triggering inflammation. I'm just gonna write inflammation right there. That's, oops, something bit me. Okay, inflammation. They can also, remember, they work as opsonins, so the fragments themselves can work as opsonins and cause opsonization. So that helps with phagocytosis. They can also cause positive chemotactic factors, release chemotactic factors, or they themselves be the chemotactic factors that like attract phagocytes, because remember, the phagocytes have to have positive chemotaxis and they have to go to the place. And these things can attract the phagocytes. Attract phagocytes, right? Another big thing that they do is they increase the vascular permeability. Yeah, they increase the vascular permeability, which is really important because, so increase vascular permeability, really important because that is related to, you may be already thinking of this, but diabetes, the word that Alessia keeps using, like pronouncing wrong in a variety of ways, diabetes, there we go. That's what I'm gonna say. I'm gonna stick with it this time, which is, you know, important because then all of these different things can squeeze through the blood vessels and like squeeze out of them and go to the actual site and work, right? So this is important, like the complement helps diabetes because it increases the vascular permeability, so things can actually go in and out. Okay, and then finally, and this is the coolest thing that they do, I like this, is they form what we call the MAC. Boom. Okay, which is just a membrane attack, not just, it's a really cool name, membrane attack complex, and that's exactly what this complex does. It attacks the membrane of these pathogens and it literally forms a hole in these membranes. Okay, so it forms a hole in the membranes. And so because of this, we have the lysis of the pathogen. Okay, so those are all the things that these fragments from the complement cascade do, and that's basically the complement. So how do we actually activate the complement? So activation is also something that we have to talk about, and it actually occurs in 3 different ways. So first way, oops, that's not number 1, there we go. First, there we go. First way is kind of like the classical one, classical pathway, which is basically the antibodies. So antibodies and your adaptive response, your adaptive immune response, they bind to the foreign substances, and they begin to excrete series of enzymes that actively, you know, that increase the activity and cause the complement. So antibodies bind to pathogens. They bind specifically through antigens of the pathogens and activate complement. I don't know why I write all of this. I could have just written antibodies there, but okay. The second type is is like known as the alternative pathway. And the alternative pathway, this is basically like independent of antibodies. So you normally have C3, which is like kind of like the beginning protein of this complement, and it's just all of a sudden it's cleaved by, it's cleaved, and then you get C3A and C, oops, why do I keep not writing? That's not a C. C3B, and then C3B kind of is activated and it causes more C3 to like cleave in two, and so C3B basically it accumulates. When I put brackets around something, that means it's concentration. Okay, it accumulates, and once that accumulates, it basically binds to microbial surfaces. It binds to the microbial surfaces, and it like there's more and more C3B that happens, that does this, and this leads to opsonization, which we all know, right? And it triggers also, so it's opsonization and it triggers the rest of the complement cascade. Okay, and then the third way is more based on basically like a component found in certain microbes. So mannose, which is basically a sugar that's found in bacteria, fungi, and like even certain viruses as well. But this mannose, it basically attaches to like activating molecules known as lectins. So this is why it's known as a lectin pathway. So mannose attaches to lectin activating molecules. And then this brings about, which equals complement cascade. So this is how we activate the complement cascade in that way. Nice and beautiful. My goodness, I've been trying to like leave space, and I think I may have figured it out, like left a little bit of space, but who knows. Okay, so then we move on to the actual processes, and the processes are going to be the inflammation, which I'm going to put here. Hopefully it's going to fit, and then the fever, which I'm going to put here. Again, hopefully it's going to fit. Processes. Okay. So processes, inflammation. So this is going to be a, again, non-specific, innate, non-specific response to tissue damage. So that's what I'm going to write, non-specific response to, oops, oh, I can just say damaged tissue, okay? And so because of this, there's a variety of things that can, you know, that come about. So there's a few symptoms that come about for this inflammation. The damaged tissue can be basically anything. It could damage tissue that can happen because of chemical abrasions or abrasions of any kind, cuts, pathogens, even like sunburns, right? This could be considered damaged tissue. So even like heat could be considered damaged tissue. So there's two kinds of inflammation. Acute, we like. Chronic, we don't like. Okay. So acute is going to basically quickly develop and be, it's acute and short, right? So it's short-lived, and this is going, so it's gonna like rapidly develop. It's gonna be short-lived, and it's usually beneficial because it eliminates any kind, it eliminates or like resolves whatever condition is, you know, whatever tissue damage is happening. So I'm just gonna write here, acute, no, not cute, sorry, quick. And short, quick and short, typically beneficial, even though it's gonna, like as we go through the symptoms now, it doesn't look like it's beneficial, but a small bout of it is going to be. It's typically beneficial, and it eliminates or resolves the issue, the underlying damaged tissue issue, the issue in the tissue. Oh my gosh, that's amazing. Sorry. Okay. With chronic, it's going to be so bad, so much inflammation that it can actually damage the tissues and even cause death of tissue. So can damage, even death, I'm just gonna write here, even death tissue. And it can basically result in, like if you have more damaged tissue, it can even result in disease. Okay. So what are the signs and symptoms of this? They're similar, right? The signs and symptoms are the symptoms. I'm just gonna say the symptoms are the same, except chronic's just gonna be prolonged, too much of it, and it's gonna be bad. But acute, we like. So the symptoms, I really have a fun way of remembering it in Italian. It's rubore, calore, dolore, and then edema. So rubore, which means redness. So we have redness. Calore, which means heat, like calor. You ever heard of like calor? Sorry. I don't know what I was trying to say. Heat, dolore, which is pain. And then, of course, at the end of it, we do have like some, like fluid kind of leaks out, as we'll see. The fluid kind of leaks out and then builds up, and that's going to cause like swelling and because edema, right? And so, yeah, edema. Okay, so localized, not too much. It's going to be acute. It's going to be good for us. That's a thumbs up. Too much, it's going to be chronic, and it's not going to be good for us. And we're going to have to actually treat it with specific things that actually like inhibit some of the molecules that allow for inflammation, that kind of trigger, you know, various things that inflammation brings about. So the first things that inflammation, where am I going to write this? Like so. Okay. So why is this important? I'm gonna write important because, so the first thing, and I'm not gonna have any space, so don't know how to do it. I'm not gonna do, it's like the blood vessels are gonna dilate and they're gonna actually increase in their permeability. They're gonna become more permeable. So increased dilation and I'm gonna write here, permeability of blood vessels. Okay, and so because of that, that's going to lead to, you know, more blood in that localized area, so redness, right, and heat as well. So that makes sense as to why you'd have the redness and the heat, right? So you have this increased dilation and permeability, which is really important because you have delivery of more blood and therefore more phagocytes, more oxygen and nutrients to the sites of infection. The veins also are more, or like blood vessels in general, are more permeable, and so the phagocytes can move into, like, they can come out, remember diaphoresis, they can come out into the damaged tissue, and then, you know, they can, like, this is me, I don't know what this is, they can fight with the invaders. They can fight with the invaders or the pathogens. They can, you know, because of this increased blood flow, they can deliver more antimicrobial chemicals. The increased blood flow can also cause fluids to leak. So the fluid leaking is going to obviously cause edema, right, which I have right here. The edema can also, you know, put pressure on nerve endings, which is going to cause pain. So this is where the symptoms come from. Pretty much this alone causes all the symptoms. We can also have, just more delivery of neutrophils and monocytes. We can have delivery of fibrinogen, which is blood clotting. So if you remember blood clotting stuff, which is found in our plasma, really important because it actually helps us form a wall around that site of infection so that the infection doesn't go anywhere else, like the pathogen and the toxin, if the pathogen has a toxin, stay localized in that area. So it's important to kind of form off a wall to prevent the pathogen and toxins from spreading. And in so doing this, sometimes pus can build up here, and so it can get pushed up towards the top, and that's where we have like abscesses and pimples and you know, zits and not so fun things that are definitely not fun. So how am I going to write all of this down? So delivery of more blood, all right, delivery of more blood, which equals increase phagocytic cells, oxygen, and nutrients to the site of infection. And then we also have like delivery of fibrinogen, which forms that clot, like let's call it a clot wall because that sounds lovely. A clot wall, a blood clotting wall, which kind of localizes the infection. And then we're going to have also, with the blood flow, more antimicrobial chemicals. This line right here was a mistake, so so I don't confuse you guys, I'm going to get rid of it with whiteout. I didn't even realize I had whiteout until now. So it's going to be any kind of mistake I make, I'm just going to, well, now. Okay, antimicrobial chemicals as well can be brought in. Yeah, and then of course the permeability of veins, it means that the phagocytic cells can move into the damaged tissue. I'm actually going to write diaphoresis because it's just, that was too much to write. So, there we go. Okay, that's really great. Another reason why it's important is because because of this increased blood flow, we have now the migration of phagocytic cells, right? So the increased blood flow delivers more phagocytic cells like neutrophils and monocytes, which are going to mature into macrophages to the infection site. And so we have, they're going to adhere first to, they're going through the blood, right? So they're going to have to adhere first to the blood vessels. Then they're going to have to squeeze through the blood vessels thanks to the fact that, and this happens thanks to the fact that we have impermeability of the veins now, increased permeability of the veins. So we have diaphoresis. And then here they're going to destroy the pathogen through, their typical phagocytic attack, right? So yeah. So should I write that? I'm just gonna write margination, which is where they attach. Like if the blood flow brings them to an area, then they're going to attach to that particular blood vessel around that area. Then there's diapedesis, so they kind of seep through and squeeze through the blood vessel, and then they go through phagocytosis with the poor little pathogen that doesn't know what's coming to it. Okay, and then of course, along with, so everything's kind of tied up to this, to the increased dilation in blood vessels, along with like, you know, the increased dilation, permeability of the blood vessels, and like in the increased delivery of more blood, there's going to be more oxygen and nutrients to the site, right? So delivery of oxygen and nutrients to site, which is just there to do what the inflammation does, right? To kind of respond to the damaged tissue and heal the damaged tissue. And so this healing of the damaged tissue is obviously dependent on what the tissue is. There's going to be areas where the tissue is really easy to heal or replace, because like things like the skin and the mucous membrane, those cells that are involved in those things, like skin cells and like epithelial cells in the mucous membrane, they replicate very quickly, hence the shedding, remember? And so those are going to be really fast to heal. But then things like the cardiac muscle or like certain parts of your brain are just very difficult to heal because, and they don't get healed. In fact, tissue says permanently damaged because they don't replicate. So yeah, like that's why like brain trauma is so bad. It doesn't replicate. You don't ever get it back. I got an email. Let's get rid of it. Okay, right? So that's basically inflammation. Crap, sorry, I forgot to tell you what triggers it. It's triggered by a variety of like chemicals that trigger the whole vasodilation in the first place. Did I write vasodilation here? That would have been good. Like vasodilation. It's a nice one word that says all of that. So this vasodilation is triggered by things like histamines and prostaglandins and bradykinins, and what else am I missing? Leukotrienes, yeah, leukotrienes, yes, so that's, yeah, yes, leukotrienes. So HPBL, for some reason, I remember that's HPBL. Okay, so those are things that triggers. So in order to treat the symptoms, if you think about it, you treat with antihistamines because histamines trigger inflammation. So you treat with antihistamines or even antiprostaglandins. Ain't that smart? I think that's smart. Or antiprostaglandins to treat the symptoms of the rubore, calore, dolore, edema. Okay, so redness, heat, pain, edema. Okay, so yeah, I think I can squeeze in fever right here. If so, this is impressive. What do you guys think? I think this is very impressive. And so this and this are going to be the totality pretty much of chapter 15. Okay, so then we have a fever. We all know what a fever is. You have a raise in your temperature, so above the normal temperature in your body, which is, I actually know it in Celsius, no idea what it is in Fahrenheit, even though I'm constantly told by my students, but I just still don't know Fahrenheit, guys, I'm sorry. But it's usually your normal temperature is about 37 degrees Celsius, so it's anything above that. And so just to talk about temperature regulation, like homeostasis for a little bit, you guys know that like the hypothalamus, which is a region of the brain, that's going to be the one that actually controls the temperature of your body. And so in order for a fever to happen, specific chemicals, known actually as pyrogenic chemicals, trigger the hypothalamus to kind of reset this like thermostat that it has, and it's actually known as a hypothalamic thermostat. And it resets it to a higher temperature. So you have that higher temperature, okay? So why do we actually like need a fever? Like, why is it good? So fevers to a small degree are good because, so I kind of talked about all this stuff. So I'm just gonna write a couple of words down, like hypothalamic thermostat. Okay, Then we can talk about pyrogenic chemicals. And these are the chemicals that actually trigger the hypothalamic thermostat to reset at a higher temperature. And these pyrogenic chemicals don't necessarily have to come from, I mean, they can come from your body actually, like an antibody and antigen complex together can basically, in the adaptive immune response, can cause pyrogenic chemicals. It can also come about from like toxins in the bacteria. So some bacteria have pyrogenic chemicals that they release. Yeah, they're sometimes released by like phagocytes that have actually eaten up the bacteria and they just need extra help. And so you're basically, the temperature gets raised because a few things start happening. You start shivering, right? You start creating thick, like there's shivering. So there's like this repetitive muscle contraction that's going to cause heat. There's going to be an increase in your metabolic rate. You're going to stop actually sweating. You're going to stop sweating. And then your blood vessels are going to constrict. And so because of all that, this is going to raise your body temperature, right? Eventually, hopefully, your fever is going to go down. And so your metabolic reactions start like dipping, they start decreasing. You start sweating. And so that perspiration actually helps you cool off. And that's how your temperature slows down, like decreases. And this is actually known as the crisis of the fever. So that's how fevers work. I'm not going to write all of that down. Should I write that down? How body temperature is raised? No, there's no space, guys. There's no space. So what does it do? I'm just going to write here, important because, kind of like what I did with not inflammation. It's important because it enhances interferon's effect. It enhances the effect of interferons, and it inhibits the growth, like the temperature itself inhibits growth of certain organisms, like inhibits growth of microorganisms because now their temperature is raised. They're very temperature sensitive. These guys, like us as well, they're very temperature sensitive. So you raise it up a little bit, it could really like stop their growth and kind of like diminish their performance. It can also enhance the activity of phagocytes. So kind of imagine as like, you know, how you heat something up and it kind of speeds things up and makes things like go faster and enhances them. This is what your fever is doing. So another one here, enhances phagocytes activity, and it also enhances lymphocytes and even the processes of tissue repair. So this right here, inflammation, it kind of enhances it, allows the enzymes to kind of work faster, processes of tissue repair. So all of these enzymes, all these cells are and chemicals are working faster just because of the increased heat. The increased heat also does the kind of like inhibits the growth of microorganisms just because it keeps them a little bit at bay for some microorganisms, obviously not all. But however, I'm going to write here, however, if it's too high, that's a problem. This is a problem because critical, like important proteins denature, right? So they get damaged and they no longer work. So it's not just the microorganisms now that are dying, but it's also your own cells and your own proteins that are dying, and specific critical or important enzymes that are dying. It can also inhibit, this is the one that really scares me. Well, everything really scares me. Okay, it can inhibit nerve impulses, and because of that, that can lead to, and I mentioned this at the beginning of the video, I think, or like the other video, hallucinations, coma, and then death. So really high fevers are not good. So guys, this is the, wow, that's a lot, right? This is the second line of a defense in your innate immunity. And I shall basically do the similar thing for your next chapter. Bye guys, I'll see you in the next video.